Well, good evening, everyone. So excited to see you guys virtually. Again, we're meeting virtually with, all the things going on in Houston. Seems like the best way to get together. We have some amazing speakers today. Randy Oliver is joining us. And from all of us on the board, really excited to see you guys. So just a couple of quick things. Here's our basic agenda for our meeting today. A couple quick announcements. Our speaker, we'll leave it open for some beekeeping Q&A after after Randy speaks with us. And then, of course, at the end of our meeting, Joel will share with us our virtual door prizes. So please hang out to the very end with us. We've got all kinds of surprises. So really quickly, this month or next couple months is actually great opportunities for you guys to get your feet wet in some classes and courses that are going on here in Texas and in the general vicinity of Houston. So the Brazos Valley Bee School is actually kicking off this weekend. It's in Bryan, Texas. And so you can check our Facebook site as well as the Brazos Valley Beekeepers Association for that school that's going on this weekend. Also, I know you guys have already heard of the Texas Beekeeping Association conference that is going to be November 6th and 7th. It's in Galveston. It's not always as close to Houston, so I highly suggest that you guys take the opportunity and drive down. It'll be fun. Plus, you can get some shrimp and walk along the sand and enjoy, some of the beach life for a little bit. I know I'll be there, and a number of our members will be there and some speaking. So please try to come and join us. We don't always get this opportunity where it's right here, basically in town for us. I know just down the road, but in town to enjoy that. There'll be some great speakers, great opportunities to connect with other beekeepers. All right, those are the biggest events kind of for us, especially in education around beekeeping. So we'll go ahead and kick it off with our speaker today, Randy Oliver. If you have not heard of Randy Oliver before, I'd be surprised. He is definitely a guru in beekeeping. He leads scientificbeekeeping.com and is really the go-to for a lot of the evidence-based research that's been going on in beekeeping, varroa management, all types of things. And so today he's here to talk to us about bee biology and what does that look like over the course of the season, and really a great opportunity. So hope you guys really learn a lot. Don't forget, we'll have opportunities to ask him questions at the end. So, with that, Randy, I will turn it over to you. I'm going to stop the share so you can drive the bus here.
All right. Well, I got a question for Joe. What's a virtual door prize? Is that something you get to see on your screen?
No, it's a real door prize. It's a real door prize. We just do it virtually.
It's a real door prize. Okay. Those virtual ones are really cheap, I imagine.
Yeah.
Okay. Hey, so beekeeping is very local. It differs everywhere. What I'm going to be covering here, let me share my screen here, is the basic biology that applies pretty much everywhere to beekeeping. And let's see here if I got, okay. And what I'm going to do is I call this reading the combs. And I'm going to blast through this. So if you have because there's a lot of 160 slides. So to get through, so I'm going to go pretty quick, and you can ask questions later on. And I hear there's a lot of beginning beekeepers there in your group. So if you Google on beekeeping, you're going to get 32 million hits back there. After you read about the first million of them, you're going to be so confused you won't know what in the world you're going to do, because there's a lot of very conflicting advice. And this brings us to the papers by Dunning and Kruger called Unskilled and Unaware of It: How Difficulties in Recognizing One's Own Incompetence Lead to Inflated Self-Assessments. So this is, I grew a Dunning-Kruger curve here for beekeeping. And this would be your self-assessment of your knowledge and ability on the y-axis, and your years of serious beekeeping experience, typical. You start off with your first hive and package bees or something, give them some sugar syrup, put them in a box, they build up, they make a honey crop. And then this is your peak of inflated self-assessment right here, where you go, wow, this is so easy. All these other beekeepers are stupid. I'm going to start my blog on how to be the best beekeeper in the world right here. And that's great. You get some people following you. And And then after a couple of years, when all your colonies die from Varroa Mite, you hit the trough of disillusionment right then. And a lot of people hang it up right here, but a number of people get it back on the horse, and they will. And this was even before Varroa. This was still common to lose hives in your first couple of years. And then you get on this slope of experience and enlightenment. And I say 3 to 10 years here. There are some first-year beekeepers who've been keeping bees for 20 years. They never learn anything. Their bees die every year. They buy a package every year. They're great customers for those of us who sell bees. They come back every year and they never learn. But many of us will slowly start to learning and slowly start to attain mastery of the craft. And if you notice this slope never flattens out. This is the exciting thing about beekeeping is I've kept bees over 50 years and I learn more every year than I have ever learned. Is that you're always going to have questions. And that's the exciting thing about beekeeping. And by the way, if you ever want the axiom is, if you ever want a straightforward, simple answer to any beekeeping question, always ask a second-year beekeeper because they're at the peak of their self-confidence in being able to answer any beekeeping question at that point. After that, it becomes much more difficult to answer questions. So I put together the rules of beekeeping from the honeybee's point of view, as opposed to a beekeeper's or human's point of view. Here's the rules. Rule number 1. They need a warm, dry cavity. When they came out of the tropics, out of Africa, where they first evolved, moved into the temperate climates, they tended to go into hollow trees for nesting cavities, protected from the rain and the wind. As far as beekeeping husbandry, that's pretty easy to provide. The bees don't give a whit whether they're in a Langstroth hive or a Warrior hive or a top bar hive or any other hive, what color you paint it. They have no idea they're even in a man-made hive. As far as they're concerned, they're still living in a hollow tree. So very easy as far as bee husbandry. Any kind of sealed box that keeps the wind and the rain out is good for the bees. Second rule, they need food and nectar. Their 2 foods are pollen and nectar. Pollen for protein and lipids, nectar for carbohydrates. Some flowers provide both, like this almond flower, they'll get both nectar and pollen. Others provide just nectar or just pollen. From a beekeeping husbandry standpoint, you have some choices. You can live in an area like a neighborhood where there's irrigation, where there's nectar and pollen all during the season, or you can do like a migratory beekeeper. When it dries up in your area, you can move them somewhere else, and many beekeepers follow the bloom across the country. Or you can give them supplemental feed, sugar syrup or pollen substitute, if it's indicated. And that's pretty much straightforward as far as that. Very easy on the husbandry on food and nectar. And the last thing is parasites. Every species on Earth has parasites, except for some parasites, but some parasites even have hyperparasites. The honeybee has right now in a coevolution with a recently introduced parasite, the Varroa Mite, pictured here on some drone brood. And most strains of honeybees, European honeybees, have not yet adapted to the Varroa Mite. Some of us are working hard at selective breeding right now, 4 strains that are, but most of you, unless you're dealing with Africanized bees down there in Texas, your European stock will not exhibit very strong resistance to the Varroa Mite. So from a husbandry perspective, since you're using a stock that you already know does not have innate resistance to the parasite, then it behooves you to help your bees, to help them manage this parasite. End of rules. Let's go back over the rules really quick in case you forgot any. They need a warm, dry cavity. They need food. They need Varroa management. That's it. Anything else anybody tells you about the rules of beekeeping pretty much are a reflection of that own person's ideology rather than what the bees actually care about. I do have a short list here. This is on my website. On the beginner section, there's a first-year beekeeping page. And this is on that page, my recipe for healthy hives. Keep young, vigorous queens. We all know that with humans, the females improve with age. That is not the case with honeybees. Young queens, typically a queen that has a fairly long laying period during the summer is going to be good for about 1.5 years before she starts running out of stored sperm in her spermatheca. And then she's normally naturally superseded by her her daughters. That's natural. To try to keep a queen longer than that year and a half you're pushing your luck. Vigorous young queens are generally much more vigorous in the colony. Provide good nutrition. We already talked about that. Control Varroa, not just at the end of the year, but all year long. The best time to control Varroa is starting in the spring. We're talking February, March. That's when you want to get on Varroa, and that will make it much easier for the entire rest of the year. Minimize toxins in the hive. Don't go dumping a bunch of junk in your hive. Humans like essential oils. Bees hate essential oils. Minimize exposure to agricultural toxins or anything else that leaves residues in the combs. And then the big thing that separates successful beekeepers from less successful beekeepers is successful beekeepers are proactive rather than reactive. They are ahead of the problem starting rather than constantly dealing with problems that have already started. So my suggestions: 1, honeybees are a fascinating eusocial insect, but so are ants and termites and yellow jackets. There's nothing that special about that. They are basically animals. So all the principles of basic animal husbandry, if you keep chickens or pigs or cattle, those same principles apply to keeping honeybees. Second one, copy consistently successful local beekeepers. The question then is, how do you tell a consistently successful beekeeper? It's not necessarily an officer of the club. They're not the people who make the headlines. They're not the ones necessarily to get filmed or make it to the magazines. Easiest way to tell in the springtime is this beekeeper buying package bees to replace his dead outs, or is he or she trying to get rid of bees because the problem is they have way too many bees and they need to either give them away or sell them, or all the colonies are going to swarm. So the easiest way to tell a successful beekeeper is those who have an abundance of bees in spring rather than needing to purchase bees in the spring. And understand the 4 horsemen of bee apocalypse. And these are chilled. Honeybees are a tropical animal adapted to make it through the winter by creating a tropical environment within a cavity. Poor nutrition. They need nutrition for immunocompetence and growth. If they don't have nutrition, they can't do either of those. Toxins, man-made toxins, natural toxins. I don't know about where you live, but we have a couple of toxic plants here in the Sierra Nevada that if the bees work them, it can really harm or even kill the colony if they work those particular toxic plants. The other big source of toxins in beehives are beekeeper applied miticides, mainly the synthetic miticides, which contaminate the combs. And then parasites. And viruses are parasites, bacteria are parasites, and our big parasite, the Varroa Mite, which is the vector for a virus. So understand, it's these 3 things that cause problems in hives. And suggestion number 4 is to base your management decisions not upon what somebody tells you or what some book tells you, but upon the biological needs of the colony. And you can tell what those biological needs are by simply learning how to read the combs, by opening the hive, pulling out a comb or 2, looking at it, and interpreting that comb will tell you almost everything you need to know about successful management of that colony. I like this quote from 1920 from Ticknor Edwards. And what he says, the best bee master is always the one who most exactly obeys the orders from the hive. You're never going to train your bees to change their natural behaviors. You're never going to change their behavior in any way. The best you can do is provide cues that make it look like you're changing their behavior. So we beekeepers who are successful, we are servants to our bees. We make their life better. And then if you make their life better, then they can give you a return on your investment and give you saleable products from the hive. So let's first understand the business model, which is the same thing as an ecological niche of the honeybee superorganism. First thing is, this is a highly efficient competitor for sporadic and limited floral resources. There's a lot of other pollinators also competing for that pollen and nectar out there. The honeybee has 2 objectives, and if you understand the 2 objectives of the colony, you can you won't be fighting your bees, then you'll be able to work with the bees. The one is to store enough food to survive dearth periods, either summer dearth, winter dearth at any time. And they do that by storing honey. That's why they're called honeybees. So always keep in mind, one of the two most important things for a honeybee colony is that storage of honey. So make it easy for them to store that honey. The second thing to do is they need to reproduce. And they reproduce by two ways, by sending out drones and sending out swarms, drones being more cost effective than sending out an entire swarm because they want to propagate their genetics. Understand this is a big issue for the honeybees, and trying to prevent them from swarming is like trying to talk to teenagers into not having sex. It's not going to happen. So honey management and swarming management are your two main management priorities. And then understand that the honeybees get their jump on all the solitary bees by keeping this standing army of cooperating foragers. They're not just foragers, but they talk to each other through the dance language, so they can do things that the other species of pollinators are unable to do. And the standing army works very intelligently, because what they do is when there's no food reserves out there, they just aren't food available. They just sit there and they just bide their time. The mid-aged bees do not transition into foraging, and the foragers just sit there waiting for the signal to the scouts. Excuse me. So first thing in the morning, the foragers don't fly out. The scouts fly out, a handful of scouts, and they check and they come back and they say, oh yeah, those floral sources are still there, go out and get them. Or they'll say, no, they're not there anymore, just hang tight. And we see this in almond pollination, where we'll take the big, strong colonies of bees to the almond orchards, which are just solid bloom. And in the morning, man, they are just foraging like crazy. And if you have a scale on the hive, you can watch the foragers all fly out with the scale. By noontime, you walk through the orchards and every hive looks like it's dead. There's not a single bee flying in or out of those hives. You pop the lid and it's wall-to-wall bees in the hives. What that's telling you is they've already gathered all the pollen during the morning. So typically the way you tell if you're pollinating, stocking enough hives in the almond orchard is you don't want to see any flight by noontime. That means they've done all their pollination for the day in the morning, and you have to wait for the trees to produce more pollen overnight for the bees to start foraging again in the morning. And all members share food. They have what's called a communal stomach in the hive. And with that communal stomach passing both the nectar or honey and the jelly from the nurse bees, they all have an idea of the nutritional status of the hive, and they all flourish or starve equally. So when a colony starts to die from starvation, all the workers start to die together by starvation. The only bee in the hive that is considered non-expendable is the queen bee, and they will feed her to the very end. So the last handful of bees remaining will try to keep her alive. Now, the queen, people often think, wow, the queen is such a special insect. She functions as the ovary and the pheromonal heart of the colony, but she has very other little influence. The special members of a eusocial insect colony are not the reproductive female, the queen. That's normal. That's the default. All other insects, all other Hymenoptera have reproductive females like a queen. The special thing about a eusocial insect are the non-reproductive females that give up their capacity to reproduce in order to put all their attention towards one single female in the hive. So reproductive fertile female, that's normal. The special ones are the bees. So the queen has very little influence over what happens in the hive. The ones who run the show are the nurse bees, and they do that because they are the only ones that produce and distribute jelly, the supply, the currency of nutrition in the hive. And they share it via trophallaxis with the queen, with the larvae, with the mid-aged bees, and with the returning foragers. So the nurses run the show. So understand, if you want to help your colony, the bees to help are the nurse bees. In colony development, so I plotted out 3 years worth of colony development. Excuse me for my drinks. I had radiation therapy a few months ago for a tumor on my tongue, and it wiped out my salivary glands. So my mouth dries up every couple of minutes, and my lip starts to stick. So I have to drink. So I got 3 seasons here, and the colored part indicates a nectar and pollen flow. And this is the colony population. And there's 4 periods in a eusocial insect colony. The first period is when food becomes available, the colony buildup starts. And with honeybees, this population curve lags about a month behind where the pollen starts to become available, or a month behind where it starts to become unavailable. They go through the buildup phase. And then when the colony is built up to about 2/3 of its maximum size, it goes into reproduction phase and raises a bunch of drones and will likely send out a swarm or 2. And then they will recover and start to build up again with a new queen, and then they go into food storage phase. Optimally coincides with the maximum peak honey flow. And then when that honey flow and pollen flow dwindles off here, then they want to eliminate as many mouths to feed as possible, and they go into this dearth phase where the older foragers fly off, and then the remaining bees hunker down as long-lived diutinous bees or winter bees, summer or winter, same physiology, and they wait it out until pollen and nectar become available again. So the dynamics of the hive population are based upon attrition versus recruitment. Attrition is the loss of workers by either being lost in the field, or a forager bee is only good for usually about 8 to 12 days of foraging, and then they're lost or their wing muscles wear out. They don't live much more than that amount of foraging. So a very rapid turnover of these foragers. And they're replaced by recruitment, the rearing of new workers. And this is the way the honeybee colony deals with most problems, by simply rearing replacement bees that take the place of the bees that get sick or get lost or killed by whatever. They just go right back into brood rearing and rear a bunch of replacements. And I made a graph right here to show the daily average change. So during the springtime, a growing colony is adding from around 500 to 600 additional bees, net additional bees per day. Now, if the queen, okay, Joe, get ready for this. I'm going to ask you an arithmetic question. If the queen's laying 1,500 eggs a day, and you have a net gain of 500 bees a day in the colony, how many bees a day are dying of natural attrition? Want to try it? 1,500 minus 500 is 1,000 bees a day are dying of natural attrition during that rapid buildup. So those beekeepers who are hesitant to sacrifice 300 bees for a mite wash to keep their colony alive and healthy aren't understanding how rapid the turnover of bees is in a colony. Sacrificing 300 bees to get an idea of the Varroa infestation rate is minimal to the colony. And then finally, the colony comes in equilibrium of the rate of attrition matches the rate of recruitment. That happens at about 40 times the egg laying rate of the queen. So a queen that can lay 1,000 eggs a day, they're going to come into balance around 40,000 bees in a hive. If she's laying 1,500 eggs a day, it'll be about 60,000 bees top population. And there's a few colonies that will even be higher than that, maybe get up to 70,000 bees. But typically a decent colony is around 40,000 bees. And then when attrition exceeds recruitment, they start losing bees at a slower rate. On my website, and I wrote a series of articles using Lloyd Harris's data. I worked a lot with Lloyd Harris. He had a huge data set from Canada from 1976, and he gave me permission to use it and colorize it and to show the age structure of a colony over the season. Again, this being Manitoba, Canada. So these are the winter bee population, the blue and purple right here. And then the spring bees are these red ones. So these are bees up to 12 days old, 13 to 24 days old, 25 to 36 days old, and very few bees live past 48 days anytime during the spring or summer. Here's your total colony population. And then the dotted line is the amount of brood of all ages in the colony. So we're going to return to this graph over and over again. What I did is I broke it down into, I think, 13 different biological periods over the course of the year. And if you're interested in more information, there's a series called Understanding Colony Buildup and Decline at my website. You can read all the articles. I'm going to blast through these very, very quickly. One big tip here. Don't do anything to hive unless you understand what you're doing. Before you even think about lighting a smoker or taking a lid off, understand exactly what you're going to be looking for, what frames you're going to pull out, what you're going to be looking for in those frames, and what if you're going to do if you see this or that in there. Have your plan ready beforehand. For a commercial beekeeper, professional beekeeper, a hive inspection typically takes about 15 seconds. You crack it open, you got the frame out, you look, bingo, that frame's back in there. The bees don't even know one puff of smoke is enough because they don't even have time to mount a defensive response. Okay, it's only if we see something amiss on that middle brood frame will we go further and look at other frames in the hive. So it's very easy to inspect quite a few hives during the day. Now that will come with experience and training your eye, but start out by always having a plan before you take the lid off the hive. The other thing that's handy is for every club to make a graph like this, a phenological graph that shows what plants are blooming at what time. That's the phenology of the plant bloom. And then overlay your typical buildup period, your swarming period, any dearths that you have. If you have a toxic plant, like we have buckeye right here, if we have a dearth here, we always have a dearth right after apple bloom also. It's typically 60 plus days from the time they initiate the brood nest until swarming, and then other 30, 60 days to the main honey flow in many areas. And again, each area is different, but if you have something like this for your beginners, it really helps a whole lot because they can then look at the phenological table and see what they should be doing at any time of the year. And I overlaid for where we live in the Sierra foothills, what different actions the beekeepers should be looking for. So this would be very handy for every club to do this, because all of my management decisions are made by counting backwards from my goal at any time of the year. So you know it takes, depending on how big the colony is, 6 to 10 weeks to build up from initiation of brood rearing to full colony strength. So you count backwards before your main honey flow. You know how long it's going to take up before they get, how many weeks until they swarm. So count backwards. You know how many weeks it's going to take before Varroa builds up too much. So count backwards and plan your strategy ahead. Okay, let's get into reading the combs. You're going to look at the honey, the bee bread, the fresh pollen, what the nurse bees do, the amount of jelly they're producing, the open brood, and the sealed brood. Now, the other name for sealed brood is Varroa food. So if there's sealed brood in the hive, assume that the Varroa mites are reproducing. And the more sealed brood, the more rapidly they will be reproducing. Okay, so I told you that the nurse bees run the show. The nurse bees only fly outside to defecate occasionally. They have no idea what's happening outside the hive. If you want the view from the nurse bees, what they see, it's this. It's totally dark in the hive. They don't see anything. Most of a honeybee's life is in total darkness, except for those 8 to 12 days at the end of its life when it goes out and forages in the sunshine. But most of the honeybee's life is in total darkness inside the hive. So they use different senses than eyes. So forget being human senses and how humans experience the world. Understand how bees experience the world. It's all about cues from outside that trigger their behaviors. And the bees monitor the amount of incoming, the nurses, the amount of incoming nectar. They monitor the larval pheromone production. They monitor the amount of incoming pollen and where it is placed in the hive. And I'm not really sure about whether they monitor the honey reserves or not. That's controversial. But these are the cues that they look for. What you as a beekeeper look for is this interface right here between the honey and the brood. Looking at this honey, because it's white looking here, that means this is relatively recent honey. The airspace between the capping and the honey has not dissipated out. After a while, that airspace will dissipate out and the capping will touch the honey, and then the honey, stored honey, will look dark like these cells right up here. It's not necessarily dark honey, it just means that it's been there for a while and the airspace has been lost. Okay, so the capped honey, now this is older honey, you can see it's lost the airspace. These are your energy stores. Any bee bread, pollen stored around the brood, is a protein reserve. And this area is your energy and protein demand, the brood rearing. Now, understand this protein reserve, this is the leftovers. This is what the nurse bees did not eat. They don't want to walk that far away from the brood to get this pollen. So this means, yeah, there's a reserve, but this really is not stimulatory to the bees. It's the pollen that's placed in the middle of the brood nest that's stimulatory to the workers. The energy interface for the hive is this line of cells right between any pollen band and the honey. And you're going to see one of two things in these cells. It'll either be honey or nectar. If it's honey, that means they're uncapping the honey and working their way up into the reserves. If it's fresh nectar, that means that they're pushing the brood nest downward and they're putting on reserves. And then here's your protein interface. Now, you don't necessarily have to have a protein interface. During the springtime, the nurses may be eating every bit of pollen, living hand to mouth as rapidly as it's brought in. And in the morning, you may not see any pollen reserve, but the colony can still be growing just fine. So the pollen does not need to be fermented for bee nutrition. The nurses prefer fresh incoming pollen, and they eat it, and they can eat it as fast as it's coming in. As you take a look here, where I'm doing a Nozema squash of the bee's guts and then diluting it in water, you can see that the nurse bees' gut contents in that water are all orangish because they're the ones that are eating pollen. If you take foragers from the entrance and do the same thing, you're not going to see that color because foragers do not eat pollen. The only bees in the hive that eat and digest pollen are the nurse bees. And then they transfer jelly through trophallaxis. And this is the informing the other members of the colony on the nutritional state. So let's go through a year, through the 4 seasons. The first cue is the presence of tree pollens. Most areas, it's tree pollens are your first pollens coming in. Even though they say they're wind pollinated, many of them, the bees avidly gather. Where I live, it's the alder trees that start blooming the second week in January. And if it's not raining at that point, boy, the bees build up rapidly on that alder pollen. You can see here in Manitoba, Canada, in March, the first tree pollen started coming in and the brood rearing kicked off right here. So here's a picture of colony in my area in early January. This is alder pollen. Now, there wasn't that much brood being fed, so the foragers can bring in the alder pollen much faster than the nurses need to consume it. So there's an abundance of this. Now, this is really good because we often get rain for a week at a time during January. And then this reserve then allows the nurses to continue to build up the colony strength in the springtime using this reserve pollen right here. Now, fresh incoming pollen stimulates the nurses to produce more jelly, to give the queen more jelly, to encourage her to lay more eggs. And the nurses then will feed every hatched egg as a larva. If there's not enough incoming pollen, the nurses then groom the brood, and they'll eat those eggs and recycle the protein. So the number of eggs, the queen can lay the same number of eggs every day. The nurses decide how many of those eggs are going to be fed and raised to pupation. The young larvae then put off a pheromone called E-beta-ocimine. E-beta-ocimine is a signal throughout the hive that all the bees are aware of that says, oh, the queen must be alive and functioning because we have young larvae. Anytime there's a restriction of E-beta-ocimine, every bee in the hive knows there's something wrong with the queen if there's food coming in. And they will immediately start building supersedure cells or swarm cells. So we'll get back to this when we get to swarming. It also stimulates, E-beta-ocimine stimulates the foragers to preferentially forage for pollen to feed these larvae. Then we get to the spring turnover. And as Al Avatable pointed out years ago, the adult bee population starts to go down as the amount of brood starts to go up. And pretty soon you have more bees as brood than you do as adult bees. You can have easily 2 times as many bees in the hive as brood as you do as adult bees in the hive. We look at it here from Manitoba. What we see is once brood rearing initiates, the bees shift their physiology from long-lived physiology, where there was almost no attrition of the bees. The population was the same. Once they start rearing bees, they shift their physiology to nurse bee physiology or summer bee physiology, and they're going to live for about 35 days. You notice 35 days later, they're all gone. All these bees, once they start rearing brood, they're all going to be gone 35 days later. During that 35 days, they need to rear enough replacement bees to be the next generation of young bees for that year. This is, and notice we have twice as many bees as brood as we do as adults right here. This is in many areas, like the East Coast, this is the most toughest time of the year for colonies. And this is where your highest amount of colony losses happen. They don't get lost during the winter. They get lost during the spring turnover because you get, once they initiate brood rearing, they can't reverse their physiology. They're going to die soon. So once they initiate brood rearing, then if you get 2 weeks of bad weather that they can't fly and bring in food, that colony is toast because they will starve. They don't have enough protein coming in to rear the replacements. And then you also have the acceleration of this aging due to foraging. So people who bring bees to almonds, if they don't have sealed brood in these colonies and all those bees start foraging, you can lose your colony population very quickly and you have bees that suddenly collapse in the almonds. So here's a picture during the early spring or late winter with this beautiful brood pattern, but barely enough adult bees to keep them warm. So these workers are the geriatric overwintered bees that are desperately trying to rear their replacements right here. And there's where we get into pathogens or parasites. So Nozema, typically a fall and springtime disease. European fabroot and shock brood, typically springtime diseases. American fabroot can be any time of the year. And then Varroa and deforming virus are late season, late summer problems. And then the paralytic viruses over the winter. So Nozema, this is a picture of 400x of a bee's gut tissue. You can see the striations of their gut musculature across here. This is a breathing tube, a tracheal tube. Every one of these white ovals is a Nozema spore in the bee's gut. This is how strongly they get infected. And they do suppress immunity. They do tend to cause early foraging. And here's a picture of me looking at a colony in February in California. We're still getting frosty nights. This is only 3 frames of bees in this colony, little tiny colony, on the verge of recovery. If this amount of brood, that's beautiful sealed brood, hatches out, this colony is on the road. It'll grow into a strong colony. But one look at this frame tells me, oh man, this colony's got a problem. Attrition is exceeding recruitment. I already said we had frosty nights. This many adult bees can never keep this amount of brood warm on a frosty night. So this tells me that they're dying faster than they're recruiting. And if these bees have a high level of Nozema, you can have these overnight collapses following a frosty night. You can see how big the colony was, the cluster of the brood was by the pollen band around here that's left around. So this was solid brood a couple of nights ago. And then during the day, the bees will come and cannibalize out all the chilled brood. And then these old bees that are left, if they have a high level of Nozema, they were not going to make it. So we saw a lot of this back during the invasion of Nozema ceranae, of colonies collapsing like this, and collapsing down this silver dollar handful of adult bees and the queen, where they had obviously a big brood nest prior. This was classic CCD. Nowadays, people, when they say CCD, that's the synonym for poor Varroa management. But back then, we actually did have this problem during the invasion of Nozema ceranae. Another confusion is that there's a misconception in the bee literature that Nozema causes dysentery. As far as I'm concerned, I have never seen one single piece of evidence that either Nozema apis nor Nozema ceranae has ever caused dysentery in honeybees. If anybody ever finds any evidence, forward it to me, but I have not ever found a single piece of evidence of any research paper that indicated that either of Nozemas caused dysentery. Dysentery is caused by water imbalance, by bees having too much water in their guts and needing to go out and fly and defecate. If they really have to go, they're not going to get very far out before it squirts right out right here. Now, if they do have dysentery and they defecate within the hive, that will spread Nozema spores if they are infected. So dysentery can exacerbate Nozema, but Nozema is not what caused it. So just because you see dysentery does not mean you have Nozema in the hive. The only way to diagnose Nozema is via microscopy. So here's a pollen grain There's a hair from a bee, typical body hair. These are Nozema spores. If you don't have a microscope, there's no way in the world you can tell whether or not the bees have a Nozema problem. And here's the interesting thing. Nozema ceranae does not normally have much effect upon well-nourished, strong colonies. Data from Canada has found it does not increase winter mortality at all. But if it's prevalent, it can slow down spring turnover and honey production. So if you have more than about 40% of the bees in the hive infected with Nozema ceranae, it will slow down the colony buildup. If it's only got 10 or 20%, then it doesn't seem to have much effect on it. It's largely associated with any time the bees are getting a bunch of protein in the form of pollen or pollen substitute, Nozema counts are going to go up. There's no reason to be freaked out. As long as they're getting a lot of protein, they can handle it pretty well. Okay, once we get past the spring turnover, we can go to the linear growth phase. And when there's lots of pollen coming in, the colonies can grow very, very quickly. This is data from Nolan from 1925, looking at package bees. And you can see they have this linear growth going up until they come into equilibrium at about 40 times the queen's egg laying rate. And then the colonies reach their peak population. This takes about 60 days from the establishment of the brood nest until they reach peak population. So that's your count backwards numbers, that's 60 days. And you can see Lloyd's colonies did this linear growth from this point to that point. And they can live hand to mouth. So this is us. We had just put second brood chambers on singles in the springtime, wall-to-wall bees down here, and they are living hand to mouth. They may have very little actual reserves, and a little bit of rain can really hurt them. Understand that there's a complete turnover of the worker population roughly once a month. Okay, if you think that your bees get to know you, they're not going to get to know you if you only inspect them once a month because it's all new bees in the hive, okay? So, and this takes a lot of protein, about 1 to 2 pounds of pollen every single week. So if it's raining, they're not getting that. So here's a colony during that buildup phase, living hand to mouth, very little reserves, but obviously being well fed, but eating the pollen as fast as it's coming in. And then if it rains, and where I live, we get lots of rain all through January, February, and March, and unfortunately sometimes into April. And it's an immediate effect on the hive. Research by Dr. Karl Kreilsheim in Germany, who placed sprinklers in the hive and then monitored the nurse bee behavior. Within 45 minutes of turning a sprinkler on, they start cutting back on the amount of jelly that they feed to the larvae. So rain outside, very strong cue to the nurse bees. And in just 2 to 3 days, they can eat all their reserves. So in Lloyd's original data, I was going to smooth these curves out, and I thought, no, let me show you the actual raw data. Look what happened here in May to the amount of brood in the colony. So I was curious, so I Googled weather history for Manitoba for 1976 when this data was collected, and sure enough, a cold rainstorm moved in right here, and immediately they cut back the amount of brood. They'll cannibalize those larvae, stop feeding them in order to save as much protein as they can to complete the feeding of the older larvae. We had a freak snowstorm some years ago during that period, and this is what the colonies looked like when you looked at them after 4 days. They consumed all their honey stores, all their pollen stores, and eaten all the young larvae. The only ones that they'll leave, only brood, will be the sealed older pupae, because this is the only, these bees don't need to be fed, and these are their last chance of hoping the colonies will survive. If they can keep them warm, these bees, when they emerge, will be the surviving bees. So this colony is in desperate survival mode. And here's a picture of me treating my bees better than I treat my own children. Because my 2 sons, this is Eric and Ian right here, they grew up with, if it's raining and snowing in the springtime, this is our bee gear. Sometimes we work for a month straight wearing foul weather gear every single day in the bee yards, because we're working in the rain and the snow, and we're feeding our bees protein and syrup if necessary to keep them building up. And I've had people say, Randy, really, you're feeding your bees when it's raining and snowing? I said, well, duh, when it's warm and sunny, they take care of themselves. If you're going to be a beekeeper, that's your job, to practice bee husbandry. And when they can't feed themselves, then it's your job to go out there and feed them. So we are very used to, we all have full sets of foul weather year, and this is how we work bees frequently during the springtime. Sometimes, like I said, a month at a time, every day. And we'll give them stimulative syrup to simulate a nectar flow also. So let's do hive inspections very quickly. Typically in the second brood chamber, the center frame will give you most of what you need to know. So if I go out to a hive for a hive inspection, give them some puff of smoke under the lid, spread the frames apart, pull out the center frame, take a glance at this, I put this back, close the lid, I'm done. 15 seconds. This look right here tells me everything I need to know. They got honey reserves, they got some bee bred pollen reserves, they have very high larval survival, no signs of disease, everything looks great, close it up, leave them alone. This is healthy brood, high larval survival rate right here. This is spotty brood. So this could indicate disease or it could indicate poor nutrition. We'll return to that later on. So if you see this in the springtime, suspect European foul brood. Later in the season, suspect poor nutrition. Here's European foul brood right here with the larvae starting to twist in the cells and becoming discolored. We're seeing a lot more of this these days, and apparently new strains that don't necessarily go away. Luckily, most of the strains still respond to oxytetracycline. I do not suggest using it prophylactically. We spot treat our hives if they do have a problem with European foul brood. And most of the time, it's not a problem for us. Also, chalk brood. Chalk brood comes from a brood being slightly chilled. And the bees actually fight it by creating a fever in the hive and heating them up. And that changes the ability of the larval tissues to hold oxygen when you heat them up. The chalk brood does not like oxygen deficient tissues. So heating them up saves them. There's generally nothing you can do about chalk brood. If it persists, just requeen the colony. And different types of chalk brood strains affect colonies differently. That's why requeening can help. Okay. That's out of order. Okay, so this is European foul brood, and this I'm seeing this a lot across the country in the springtime, hampering colony buildup. So any colonies that are not growing during the springtime, that means typically either a Nozema problem, which you need a scope for, or European foul brood. And you may have to look through several frames to actually find symptomatic larvae. Generally, if you have mixed pollens from different sources, that's good, indicates good nutrition. Okay, what we do is we monitor the amount of jelly that the bees, the nurses, put around the second instar larvae. A well-nourished colony, they will cover the bottom of the cell wall to wall with jelly. So this is a well-nourished colony with the larvae swimming in jelly. So this would be, this may be a first instar, maybe a second instar larva right here. But look at all the jelly around the second instar larvae right here. When your colonies look like this, you are the world's best beekeeper. You could do no wrong. It'd be hard to kill that colony because they can handle anything because they're recruiting so quickly. Here's a colony that's only giving enough jelly to the larvae just to barely supply the nutritional needs. So these larvae will grow into being a normal looking adult bee. That adult bee's behavior will change and they will start foraging at an earlier age. So the colony will not grow as fast with larvae that are reared under these conditions. Another look for you here. And I'm showing you several of these pictures here so you can see the difference between larvae that are swimming in jelly and larvae that are hungry. This has really changed our beekeeping, learning how to glance at a frame, tip it to the light, and see the nutritional status of the bees yourself. You don't have to have anybody else tell you. There's larvae that are barely being fed enough. So if you're curious about whether it's worthwhile if you eat pollen sub, the amount of jelly tells you everything. And again, there's times of the years where you may not want them rearing brood, because when they're rearing brood, they're rearing Varroa. So for us in August, no bees reared in August will ever make it to be winter bees. So there's no reason for us to rear brood in August. We don't stimulate brood rearing in August. Come September, right now, we're out feeding truckloads of pollen stuff because now we're trying to build up our colonies for the wintertime because we are almond pollinators and we get paid by how many frames of bees we have on February 15th. And that number is dependent upon how strong our colonies go into winter in November. Okay, back to springtime, swarming impulse. Colony is full here, but look at this. We start to peak out on the amount of sealed brood, and this is when we get this swarming impulse. The colony is ready to reproduce. They want to have as much sealed brood as possible so it can recover quickly when half the bees fly away with the queen, half the workers, leaving all this, again, we have almost as much bees as brood as we have as adult workers. So you can lose half of these workers and it hardly sets the colony back at all. The swarming impulse really gets cued by or triggered by the cue of backfilling, where the brood nest starts fighting the incoming nectar and starts getting squeezed down, and there's no place for the queen to lay eggs. When there's no place for the queen to lay eggs, that means 4 days later, there's no E beta ocimine, and every bee in the colony realizes that they've run out of room in the cavity, time to reproduce. And they start rearing queen cells. So they trigger it when the bees feel that they've filled the cavity and packed the brood nest. There's nothing else to live for other than reproduction. So you want to avoid them ever getting that cue by giving them room above, either by reversing the brood chambers, which will actually cause them to tear down the queen cells and start growing again, or by adding frames of not foundation. They don't treat foundation as room at all. It has to be drawn comb in contact with the brood area, preferentially above it. Drawn comb added below the brood will not prevent swarming. It has to be placed above the brood. I got data to show, strong data to show that. If you start seeing all the queen cells being made on the bottom of the frames, now it's too late. It's time to make splits. And this is, we set up production lines. My sons now, they're making about 150 splits every day during the springtime. And we make about 3,000 5-frame nukes every springtime. We sell about 1,000 nukes right here. Here's a big tip for you. If you're ever going to be doing this, don't work where the parent colony was. Pick up the boxes of bees, move them away from the parent stand. Any bee that's going to sting you will fly back to the parent colony, and the poor guy working over here has all the hot bees right here. We can work over here without any gear on, going through hundreds of hives because there's no bees that are going to sting us right here. Big tip for you. And then this is colony reproduction. This is the last thing you want to see. Number one, you've lost half your field force. Number two, you've now got a competing colony out there competing for nectar and pollen. And number three, this, if they're successful in finding a cavity and building up, they're going to become a mite disseminator later in the season and bite you in the butt with the mites coming back to your hives from this unmanaged colony. Okay, so now when the pupae are in the sealed brood, they're packed like cordwood, very, very tight together. Notice that it takes that one adult bee covers 3 cells of sealed brood. That means that when this hatches out, a frame of sealed brood in a few days will be 3 frames of adult bees over a frame. So a 3-time expansion. That's why these colonies, when they swarm full of sealed brood, these 3 frames are going to hatch into 9 frames of adult bees in a few days. So the colony can very quickly recover its population. And they want to do that by the timing for the main honey flow. And our goal, and the colony's goal, is to reach peak population right at the start of the main honey flow. So you count backwards from here, and that tells you how you should be building your colony up. The other thing to understand is that larger colonies proportionally are more efficient at storing honey. So what we do is we make a lot of splits in the springtime. When the honey flow starts, we combine our smaller colonies into larger colonies. You'll make more honey with a smaller number of large colonies than you will with a whole lot of small colonies. And then the honey flow starts. We call this nectar shake. Pick up the frame and you shake it out. Okay, this is from Blackberry. Or this is at 10:30 in the morning. This is all nectar they had collected that morning already. And that nectar forces the brood nest downward. So this is a cue for the colony to forget about brood rearing, make room for honey storage. Remember, the main 2 things for bees are to store honey and to swarm. So we're past swarming. Now we're going to make room for honey. And when they run out of room, they will start producing white wax, which says, this is your signal to the beekeeper. 1, your colony is out of room. If you don't do something, they're likely to swarm. 2, it means this is the only time of the year you can draw a foundation. If you're not seeing white wax, you're wasting your time putting foundation on the hive. There's no magic about drawing foundation. If you don't see white wax, they are not going to touch foundation. If there's white wax, they will draw it very, very rapidly. Time to draw foundation. One thing, if you're going to put a box of foundation above the brood chamber, if there's a honey band here, that inhibits the drawing of foundation. Try to move frames around so there's brood right up to the top bars here, and then the bees will hop on that foundation better. Then for the honey flow, you need about 1 mid-age bee for every forager bee. And there's all kinds of feedback loops for that. And you can read my articles if you want to go into detail on that. But the colony has ways of balancing the number of mid-age bees against the number of foragers. And brood rearing is deprioritized. Look at this. Look what happened in Manitoba. Look at this drop in the amount of brood in the colony. What this is from is the main honey flow starting. Any bees, any brood that's reared at this period of time are not going to ever be old enough to help with the honey flow. There's going to be extra mouths to feed. So the last thing the colony wants to do is to rear a bunch more hungry mouths during the honey flow so you have more to feed out here. So they will deprioritize brood rearing. And they'll just fill the brood cells full of nectar. What the beekeeper does then is they expand the cavity by adding more boxes of drawn comb right here. Here's Tim Ives in Indiana, and he makes 350-pound honey crops out here by creating a big cavity. So the bees now have the cue that, wow, we have not filled the cavity. We should not swarm. We need to fill the cavity because we have all this drawn comb up here. And then we have the late summer population decline. No pollen coming in, the bees go into survival mode. So this is your typical pollen dearth, and the colony population drops down. And attrition, now you have a negative colony population here as they drop down their population. Now these numbers aren't as big because the queen's not laying as many eggs. Or maybe I shouldn't say that. The nurse bees are not allowing as many eggs to be reared to maturity here because they do the brood grooming. There we are, eliminating the extra mouths to feed. And if you look at your brood patterns now, you may see the fresh honey from the nectar flow. You may even see some nectar still coming in. No pollen. If you got no pollen, then those nurses are going to be eating the eggs as fast as the queen lays them. So this is an indication of protein deficit. So nothing to freak out about. This is the natural behavior for the honeybee colony to go into the dearth following the honey flow. They don't want to raise a bunch of brood. So unless you're a California almond pollinator or a or package producer where you want to have big colonies early in the season, just let them go downhill. You don't have to feed pollen. Now, those of us who do want to rejuvenate our colonies and shift the age structure from old bees to young bees, we will put on pollen sub, and the nurse bees just hop right on there, and they convert this pollen sub into jelly, and then that jelly into new bees in the hive. Oh, and then we get to our friend, our new parasite. I do have a free model out there you can learn to use to figure out your management strategy. And what happens is if you do mite monitoring with a mite wash here or a powdered sugar shake, the mite population slowly builds, but your sugar shake or your mite wash, we use Dawn detergent now, will show it growing incredibly fast. Not so much because the mites are reducing fast, but because your population of adult bees is going down. So you have a higher number of mites per adult bee. And this is where the viruses kick in. You may also start seeing Varroa sensitive hygiene, where the bees start chewing out infested pupae. All brood, this is the main behavior in most populations that are developing resistance, this uncapping and recapping behavior. So in my selective breeding for resistant bees, I'm seeing a lot of this uncapping, recapping behavior. And then you start to have a virus epidemic. The deformed wing virus, you may see symptomatic bees with deformed wings. You may see the larvae and pupae starting to die from the virus. And the higher, this is your mites per 100 bees on the red. As that starts increasing, you can see deformed wing virus and the paralytic viruses increase concurrently. So the trick is try to keep your mite level down in the hive to this low level all year long. When you let it go up, then the viruses get ahead. Just like our coronavirus pandemic now, you get an epidemic of deformed wing virus in the colony. Once you get over about, there's a red indicates what percentage of the pupae are infested by a mite. Once you get past about 30%, a third of your pupae infested, you can write the colony off. You're not going to be able to save it. And you get the crash indicator. And you're going to start seeing this. The bees are unable to emerge. If they do emerge, they have deformed wings. Parasitic mite syndrome, where everything just starts to look really, really ugly right here. And again, parasitic mite syndrome, the most common cause of colony losses these days. And the telltale sign is these white fecal deposits remaining in the brood nest from all the mites reproducing in the brood. In the white fecal deposits. And for the first year beekeepers who come back after buying nukes from us the first year, and they come back and they say, oh, my bees absconded, or my bees swarmed, or my bees died from pesticide poisoning or whatever. And I said, let me take a look at your combs. You see all these white spots? This means you didn't manage Varroa adequately. And they go, oh, really? I go, yeah. So they better luck at this next year. The problem is, look at the Varroa mites on these bees right here. And if they're, you can see them on top, you know there's a lot more on the bottom. These mites drift to other hives carrying those viruses, and mite drift is a real thing. So try to get all your beekeepers to cooperate to do Varroa management so you don't have this mite dissemination from hive to hive. For you beginners, the most common form of mite management practiced by beginning beekeepers is called wishful thinking. And that's never, ever worked. So just let that go. Wishful thinking does not control Varroa mites in any way. Best thing to do is control Varroa early, starting in the springtime, and keep your mite infestation rate down all season long. There's research that I've done now recently. If we can get extended release oxalic acid registered, it's going to be a game changer for applying that early in the springtime, and just fantastic. In our operation, this is what we do. We bring our bees back from almonds. We split them 4 ways, do an oxalic acid dribble with an induced brood break, reduce the mites by 95%. While the honey's still on in July, we put a single formic treatment on to knock back the mites a bit. And when we get the honey off, we do a time-wall treatment, then feed protein to build them up, and then give them an oxalic acid treatment early in the winter. We've done this years and years. We haven't used a synthetic miticide in 20 years right now, and our colonies are healthy all year long with very low mite levels all year long. We're also doing selective breeding. We're looking for the heroes. Zeros are heroes. This is the mite count in July, mite count in August, mite count in November, mite count in December, or September, November. We're breeding only off of queens that have mite counts that look like this throughout the year. That without any mite management, they can keep the mites down to 0 or close to 0 all year long. So that's for the last 5 years. It's the only queens we have grafted off of are those that manage the mites. Unfortunately, it's not that heritable yet. It looks like we may be making a breakthrough this year and perhaps have locked this into some of the female bloodline genetics, which is the critical thing. So we'll see what happens this winter, but we're guardedly optimistic. Okay, then autumn pollen flow starts, and the bees will often store pollen below the brood nest for winter stores there. And you can see this is the bees are now being this last round of brood rearing, September, October, on the ones that will become your winter bees. And this is your worker survivorship, very short during the summer, but the ones that emerge in the fall, they have an extended survivorship over the winter until they start rearing brood, and then they change their physiology right back to springtime bees and have a very limited warranty. From that point, they die off very quickly. That's that spring turnover happens at this time. So in the late season, the first thing you want to do is control the mites to stop the virus epidemic, and then if necessary, feed protein to stimulate that late season brood rearing. Any colonies that don't take their pollen patties, we just close them up and walk away from them. They're not worth the effort. There's something wrong with that colony. Okay, so then the first frost comes, no more brood rearing. You can see they dip down to no brood, and November is typically the least amount of brood rearing worldwide in in honeybee colonies. They may do a little bit of wintertime brood rearing, but this is when the fall pollen flow ceases. And these bees that emerge when there's no other brood around, no young larvae, they, instead of going on to nursing, they just stock up, as similar to nurses, store their food in their fat bodies, develop their hypopharyngeal glands, and settle down with storage proteins to make it through the winter. Here's a forager bee, no fat bodies, a winter bee or nurse bee, heavy on the fat bodies in their abdomens. Then we get the fall turnover, where about half the bees, typically in my area around November, fly away and they die outside the colony, they're old age. So it's common for our colony population to drop by about half in November. The bees that are left are going to become these long-lived winter bees here. So instead of dying off quickly, they have this winter cohort here. Now, if you lived in probably down in Texas or Southern California, you don't get this winter bee cohort. You're going to stretch this graph out this way. My model, my Varroa model, you have options for doing that. That will show that. Then the winter cluster, it looks like a solid ball of bees, but it's actually a mantle, a closely packed bees and a loose warm area in the center. If you give them a puff of smoke, you can see that mantle outside and the bees in the loose area will move away from that smoke. And they actually make an airtight shell. They can change the atmosphere inside here. The carbon dioxide level goes way up. The humidity goes down in here, and they can keep this warm with the bees on the outside around that 55 degrees range on the outside. Most of the moisture gets ventilated out of the bottom of the colony. The best thing you can do to help your bees is put an insulation over the top of them. And that's, you can see the heat loss here by lack of insulation. A piece of insulation on top will help these colonies quite a bit. Now you have these long-lived bees all throughout the winter. No protein reserve. So this is your last round of bees that will become the winter bees. All right, I don't know if you guys have a strong dearth. Here in Northern California, in August, we have winter bees. The colonies, if you have Russian bees up here, they look like they're queenless. The nurse bees stop rearing larvae. The queen's still in there, but there is no brood in the colony whatsoever. So you can have a dearth during hot times, just as well as you have dearth during cold times. Now, in the midwinter, you may get some initiation of brood rearing. This seems to be a function of moisture balance, but there hasn't been enough follow-up research on that. And then we in California will stimulate this late winter brood rearing in preparation for almonds. My friend Keith Jarrett feeds huge amounts of pollen sub to his bees in January. And in February, when other beekeepers are desperate for bees to fill their pollination contracts, Keith's out there shaking bulk bees and selling them to other beekeepers in early February for them to boost their colonies. And we're back to the first pollen, early spring brood rearing right here, and brood rearing starts again, pollen coming in, and we're back to the beginning of the cycle. And that's it. There's my recipe on my first year of beekeeping page, and there's my website right there. Let me see, how am I? There's no clock on my computer here. Let's see, what time were we doing? Wow, I didn't do bad. Right at an hour. How's that?
That's perfect. So members on the line with us, if you're interested in asking some questions, please place those in chat. And Randy, the first question from the group comes from Joseph Lothy. And the question is, what are your thoughts on the Bee Gym by Stuart Rowith? Also, do you recommend Varroa hygienic sensitive bees in the honeybee family? And last question, there's a couple in chat. Do you have a Discord channel? What's a Discord channel?
That's my question. I don't know what a Discord channel is.
Okay, good.
Okay, so as far as the Bee Gym, I don't have hard data on it. So if I don't have hard data, I don't have opinion. Okay, as far as the Varroa sensitive hygiene, I think that was, Varroa sensitive hygiene is good for brood diseases. Okay, so for American fat brood, European fat brood, it's definitely worthwhile breeding for. for Varroa management, what we're seeing is the uncapping, recapping behavior, or specifically Varroa sensitive hygiene rather than freeze-killed brood hygiene, which is the thing to select for. So the freeze-killed test does not correlate that well with Varroa sensitive hygiene.
Okay. Thank you. All right. Kyle's question. Do you have any recommendations on methods to collect blooming pollen data for our area? So back to your earlier graph where for your area where you see, what is the pollen throughout the year? How would you suggest us doing that?
The easiest thing is I've taken photographs of bees working pollen on every flower in our area to catch the color of the pollen on their legs in their curbicula. So that's the easiest way. Just microscopy, you can do it with microscopy, but it is much more difficult, I'll tell you right now. Some pollens are pretty easy to identify, but unless you're really handy with the microscope, that's hard. The easiest way is by color, by going out to the flowers saying, hey, do I see this color pollen coming into the hive at the entrance? And that'll give you an idea.
Okay, basic observation tools.
Exactly.
Sounds good, I like it, it's very practical. All right, the next question is, what are you working on for new Varroa management methods? Did the cellulose strips you were working on not work out? Question?
No, well, hang on, let me answer that one.
Sure.
Yes, the cellulose sponges worked out very well. They're kind of pricey. So this year we tested an off-the-shelf industrial absorbent pad called Maximizer pads. They have two weights of them. We use the lighter weight one. Very, very inexpensive, much less than the cellulose sponges, and they work just about as good, and they handle about the same. So I'll be publishing the data on that. What we found out is we also did a trial on the total surface area that you need to get really good mite management. Last year we had used one half sponge or 2 half sponges. We got decent Varroa control with 1/2 sponge, the Swedish sponges. We got better with the 2 half sponges. So this year we cut them in quarters. We did a 1/4 sponge, 1/2 sponge, 3/4 sponge, or a 4 sponges, and 4/4 sponges was the best. And I'll be presenting data showing that. So we now know it takes about 60 square inches of surface area of substrate in the hive, whether it's placed on the top bars between the brood chambers or hung over the frames in a single. It takes about that much to get adequate oxalic acid transfer onto the bees.
Great, thank you for that. We look forward to seeing the published results.
Yes.
All right, next question from Perry. I just captured my first wild swarm. Should the queen be replaced, or do I assume the swarming queen is young?
There's no shoulds in beekeeping. Okay, everything's, I wouldn't make any assumptions. At this time of the year, you get a lot of smaller swarms of queens that are about to be superseded. And with the Africanized bees, they call them usurpation swarms, where if you're a bee colony and your queen is getting old, what's the best way to get your genetics into the next generation? You send out a bunch of kamikaze bees with that queen, and you take over a colony that's already got honey stores, leaving a daughter of that queen behind. You can then double your genetics in the population by taking over another hive with the usurpation swarm. So this is common, very common with the Africanized bees, but I've seen it with the European bees forever. So it's not just an Africanized trait. So that may be what you're seeing. So it may be an older queen. Take a look at the queen and see how fuzzy her thorax is. If her thorax is polished, she's probably an older queen. If her thorax is still fuzzy, then she's likely a younger queen. And in your area, if you live in an area where you have Africanized bees, she's probably an Africanized queen.
Yeah. Yeah, and we do our versions along the spectrum, right?
Right.
You know, we love them too, because they do produce honey quite well. You just have to be careful how spicy they get.
Exactly.
All right, this next question comes from Vicente, Any advice for a new beekeeper? I bought a beekeeping kit and I read that I should wait until winter to buy bees in the spring. Is November a good month to purchase bees for the spring?
That would depend upon your area. For example, I have a friend in Southern California who runs a bee supply business. And everybody wants to buy nucleus colonies in the spring, in April or May. Their dearth period starts in June. And there's nothing for the bees to eat from June until November. So the worst time of the year to start with a new colony in Southern California would be in April or May. The best time would probably be in November when they get rain and they have a pollen flow. So ask experienced beekeepers in your area when you have your spring buildup period, and that would be the best time to start.
Yeah. So I could probably answer that, and probably a couple of other beekeepers here, Vicente. So yes, you would want to purchase for the spring. We do see colonies start to build up even as early as February to get ready for the spring honey flows. But we do see dearths in July and August. So you have to watch them to see how well they do before you hit that dearth period. Would anybody else like to add to that?
I just sent a chat on that subject
Perfect.
about our mentor-mentee program.
Oh,
Wonderful.
yes.
Your local beekeepers, your local experienced beekeepers who don't have an agenda, which means they are not usually the loudest beekeeper around. You want to look for the quiet beekeepers who've been doing it for a long time. and they're a huge source of information for your local conditions.
Yeah. Luckily, as Randy mentioned, we don't get usually we don't get cold winters, although this last winter was unique.
Oh yeah, we heard about that.
Yeah, so we'll see a couple different of those spikes throughout the year of root of the colonies building, then it gets hot and there's not a lot to eat, so it goes back down, and then it gets right back up during the fall. You'll see a lot more growth usually in the fall if we don't have weather that keeps them from harvesting nectar. Yeah. All right. Any other questions? This is your opportunity, the famous Randy Oliver. All right. I think, Randy, that is it for today.
Wonderful.
But just from all of us, thank you so much for joining us today and sharing your wisdom.
My pleasure.
And all the work that you do, all the research, evidence-based practices. As you mentioned, there's all kinds of opinions.
Yes.
It's also really nice to, be able to see the data and make our decisions around.
Absolutely.
That.
And that, in life, that is just, I wish everybody would do that. Look at the data. People say, look at science, forget science, look at evidence and data. And that's the best thing. And it's my honor to be able to help the beekeeping community with that. And I appreciate everybody's support for that.
Yes. Thank you so much. Great work. Carry on. We'll be watching.
Will do. Good night, everybody. Happy beekeeping to you.
Good night. Thanks, Randy.
You bet.
All right. So next up, of course, is our virtual door prizes. They aren't virtual prizes, but they are hosted virtually. So just so we're clear.
They're real prizes, yeah.
They are real prizes, I promise. So I want to turn it over to Joe.
Okay, so we have 3 prizes to choose from tonight. We have a hive tool donated by Texas Bee Supply. We have an HBA gray, a Houston Beekeeper Association gray t-shirt, size large. And we have a Beekeeper baseball hat. So our first winner is Bobby Brown. So Bobby, if you can tell us which prize you'd like to get. Hopefully you're still online.
Hi, yeah, I'm online. I'll take the hive tool if it's available.
Okay, you got it.
Thank you.
All right, second prize winner. And this is what Nicole, after volunteering for years with the club, now she keeps winning. So Nicole Burgers, you won again. Burgers.
Yeah, that's one of my special superpowers. I do that. I'll take the t-shirt.
Okay, you got it.
Add to your
And
collection.
then, yeah, add to your collection. And then the last final winner is Joe Brown. Joe
Hey,
Brown, you still
Joe.
there?
I see
Yes,
her
she's
on.
here.
I'm here. I'm here. How exciting.
Okay, so you won a baseball hat.
Yay. Good job.
So I'm going to send you guys all a email or something, and we'll figure out how to get them to you, or you can pick it from my house or whatever. Worst case scenario, where we haven't had an actual board meeting to talk about it yet, but hopefully maybe in October we'll actually meet in person again, and I can give it to you at that point if that's the worst case scenario. So
Yes. Good deal. Well, congratulations, everyone. Love to give away goodies, especially, you know, hats and shirts from HBA. So our next meeting is scheduled for October 19th. Of course, every month we kind of revisit where are we in the Houston community and can we get together safely, or if we should continue to postpone. So we will keep you updated as that happens. And of course, our last meeting will be in November. We do not meet in December normally. So just a little heads up for where we're headed. And so enjoy the next couple of weeks, and we look forward to seeing you in October. Thank you all. Happy beekeeping.
right.